The human genome is a vast and intricate blueprint. It holds the genetic instructions for life. Imagine, if you will, a library filled with volumes of information. These volumes contain the code for building and maintaining a human being. This code is written in the language of DNA. Genome sequencing was a groundbreaking achievement. It allowed us to read this code. We can now identify the genes within our DNA. This knowledge has revolutionized our understanding of human health and disease. However, the genome is only a starting point. It's like having a list of ingredients for a cake. You know what's in it, but you don't know how those ingredients interact. The genome doesn't tell us how genes are expressed or regulated. To understand the complexity of life, we need to look beyond the genome. We need to study the proteins that genes encode. This is where proteomics comes in. Proteins are the workhorses of the cell. They perform a vast array of functions. They transport molecules, catalyze reactions, and provide structural support. The human body produces thousands of different proteins. Each protein has a unique structure and function. Proteomics is the study of these proteins. It aims to identify and quantify all the proteins in a cell, tissue, or organism. Proteomics also investigates how proteins interact with each other and with other molecules. Think of the genome as a parts list. Proteomics, on the other hand, studies the actual players in the game of life. By studying proteins, we can gain a deeper understanding of how cells function in health and disease. Studying proteins is a daunting task. The proteome is incredibly complex. It is constantly changing in response to internal and external cues. Proteins are dynamic and can be modified in countless ways. Scientists use a variety of techniques to study proteins. Mass spectrometry is a powerful tool that allows us to identify and quantify proteins in a sample. Protein microarrays are another valuable tool. They enable us to study the interactions between proteins and other molecules. Analyzing the vast amounts of data generated by proteomics experiments is a challenge. Advanced computational methods are needed to make sense of this information. Despite these challenges, proteomics holds immense promise for improving human health. By understanding how proteins function, we can develop new drugs and therapies. Proteomics can also be used to develop diagnostic tools for early disease detection. Imagine a future where we can detect cancer at its earliest stages, or predict an individual's risk of developing Alzheimer's disease. Proteomics has the potential to revolutionize medicine. The field of proteomics is still in its early stages. However, rapid advancements in technology are accelerating our understanding of the proteome. With continued exploration, proteomics will undoubtedly unlock new frontiers in biology, medicine, and beyond.